Hello and welcome once again to my channel. We're looking at AQA GCSE biology microscopy. So we're looking at the microscope and different parts of the microscope. We're going to calculate, measure cells and calculate image of cells, etc. So the first question is about the structure of the microscope. Now there are many other parts to it, but in this lesson we're only looking at 3 parts. The first part of the microscope that we're looking at today, A, is the adjustment knob. You can have fine and coarse adjustment knob as well. And B is the mirror, though the more modern light microscopes have a light bulb instead. Then you have the objective lens at C. And then in terms of their function, the objective lens, it helps you to magnify the object you're studying, and the mirror focuses the light in the observer's eyes. Then the student prepares the slide to view under a microscope, and the question says, why was iodine solution added to it, to the layer of onion cells? And it is to stain the cell. Why was a thin layer of onion cells used? And it was to be able to get light to pass through it so you can observe it. There are risks associated with this experiment, and so in order to avoid these risks, you would use the hazard, being careful with the sharp knife and using a cutting board when cutting the onion. Now, next one is you measure the cell. Measure in centimeter, but you need to convert to millimeters, so you times that by 10. Then you need to convert that 49 millimeter to micrometer. You times that by 1,000. Then the question says, calculate the real size. So in order to do that, it will be the image size divided by magnification, which is times 400. When you divide, you get 122.5 micrometer. Next section, how would the onion cells look different if they were seen under electron microscope? And it is that they would look bigger and you would see more subcellular structures. And then when you draw microscope images, you must complete the cells and you must say what the magnification is. Was it times 100? Was it times 10? Was it times 40? You need to be very clear. Thank you.